So hello everyone, I'm very happy to see many of you from various different places and various stages of my life joining us here today. Today I'm going to talk about my PhD project, which was about life history evolution under cancer risk. And so this thesis was structured as follows. So I had an introduction and then a preface that was intended for a broader audience. And then I had three scientific chapters and a discussion. And today I'm going to start with introducing you to the topic with this, based on this introduction and preface I've written. So all of you would be familiar with cancer, and you might be thinking of it as a disease, human disease of all age. And with this, you wouldn't be wrong. Indeed, cancer incidence in humans increases with age, and it is an important human disease. It actually just overtook cardiovascular diseases as the leading cause of death in high-income countries. And this might be the extent to which you thought about cancer before, and this was definitely my case until I started this PhD position. But today I want to talk to you about why cancer is a problem across the tree of life and for all the multicellular organisms. And for that, I would like to take you a bit back in time to the origins of life. And it started in unicellular ancestors. These unicellular ancestors were having cell divisions to reproduce themselves. So once there was a transition to multicellularity, which occurred multiple times over the macroevolutionary time, there needed to be some cooperation between these cells. And this meant that some of the cells had to forego their own reproduction to contribute to the reproduction of the organism itself. So how is this ensured? So there is some genetic mechanisms in the cells that make sure this happens, this cooperation is maintained. And these are related to these so-called foundations of multicellular cooperation. And today I'm going to talk about two of them to you. So one is inhibition of cell divisions, which I mentioned before. And the other one is the programmed cell death, which kills the cells that have accumulated some mutations that might have some DNA damage problems. So I already alluded by this that there might be some DNA damage that can happen and disrupt these mechanisms. And if these mutations, this DNA damage happens, this cooperation mechanisms might be disrupted. And these disruptions might lead to uncontrolled cell divisions as well as evasion of cell death by some cells. And these are things that are associated with cancer. And with this, I think it is clear that cancer is not only a human problem, but is a problem that is faced by all multicellular organisms, from vertebrates to invertebrates. And even if you define cancer as uncontrolled growth, this is even seen in plants. And to look at one of these examples, for instance, in Hydra, we see that this is a very simple animal with very few cell types and very few cells. And even in this one, shown in right in here, there are some uncontrollable growths happening. And now if we go back in time from extant organisms to extinct organisms, when we look at fossil data from dinosaurs, we see the evidence for bone cancer in dinosaurs, for instance. So I already talked about this. Mutations might happen, that they can disrupt these mechanisms that ensure cellular cooperation. And these mutations tend to happen in during cell divisions. So this implies that more cell divisions an organism has, more mutation risk it will have. So this might mean that all else being equal, size associates with cancer risk, and cancer risk increases with body size. So is there evidence for this? Well, let's look at within humans. And here I always show this example where I compare a woman of 10 centimeters shorter with myself and say that 10 centimeter increase in height associated with 16% increase in cancer risk. And it's not only humans that have this kind of association within species. For instance, in dogs, it's been shown that larger breeds of dogs have higher cancer risk. So this 10 centimeter of increase that was associated with 16% of more cancer risk is actually quite minor when you think about the diversity of body sizes across the tree of life. So with this association, you might expect that elephants to be plagued with cancer and succumb to cancer even in early ages. But this is actually not the case when we look at the data. So data actually look like this. So in the x-axis, we have here mass and lifespan in log scale, and in the y-axis, the percent of necropsies from zoos with cancer incidence. And what you can see is there is no linear increase with body size, and this regression is actually quite flat. So this is something called Piro's paradox. And there are various mechanisms that researchers propose that might resolve this paradox. And one of these is that large-sized organisms can have some cancer-related adaptations in the cellular or tissue level to reduce their per cell division cancer risk. And there is some evidence for this as well in empirical studies. For instance, researchers took fibroblasts from humans and elephants, and they irradiated them. And what they observed was elephant cells were killed faster than human cells. So this kind of means that elephant cells are more vigilant and they're less tolerant to DNA damage compared to human cells. So DNA damage response is one aspect of this. And there's also some genomic evidence for that. It's been also shown that elephants have 20 copies of this gene, TP53, which is one of these genes that have a big role in cancer defenses. And this is compared to humans who have only one. There are other organisms such as naked mole rats who have different ways of dealing with cancer risk. And naked mole rats are an interesting example because they famously have also very low cancer incidence. And they owe it to this mechanism called early contact inhibition, which I'm not going to go much into detail today. As such, there's this burgeoning field of comparative oncology, and researchers are looking at different evidences for different mechanisms in the genomes of long-lived and large-sized organisms, and discovered that there are various mechanisms that these organisms might be using against cancer risk. And with that, I will start with my first question, which is, and ask that, how are cancer defenses optimized across body sizes? So this is my first scientific chapter, so chapter 2. So with these cancer defenses, what I mean is cell-level strategies for growth and cancer suppression. So we implemented this as 7 traits that make part of one strategy. And these traits are related to DNA damage response, as I showed before, but also telomere length, which is related to how many times a cell can divide, as well as cell division and differentiation. So then what happens in this model is that the organism starts its life as a zygote, and using this strategy that is composed of these 7 traits, it grows to its target mature size and maintains its size through its adult lifespan. And during this time, it faces different death risks, including some that are related to stem cell management and somatic maintenance, but also to cancer and extrinsic mortality. And then I'm not going to go into more of the details of this model, but what we did was to do numerical simulations and use a genetic algorithm to find the optimal strategy for a given body size. So with this, I'm going to start with the results. So in the x-axis here, I'll show the target mature size. In the y-axis, there is one of the traits that make part of this 7-trait strategy, which is number of differentiation levels. So how many differentiation steps it takes from a stem cell until the terminally differentiated cell. So what we've seen is in this trait and in some of the other traits as well, there is some size-specific adaptations. So you observe some sort of pattern with size. And here, for instance, number of differentiation levels increase with body size. Whereas the pattern is less clear and not there actually in some of the traits. So these traits seem less important for management of growth. But when we dug a bit deeper into this, we've seen that at least in some of these traits, there were some co-adaptations. So what I mean by this is, for instance, one trait was increasing and the other one was decreasing. And there was this like, they were correlated with each other in a sense. So this kind of indicates that there might be different ways and different combinations of these strategies to achieve the same body size, same sort of cancer defense and growth. So now I come to the more exciting part, at least for me, of this project, which was about taking the small adapted strategies and using them to grow a large body-sized organism, and vice versa, taking large adapted strategies and trying to grow a small organism. And here I will show in the x-axis the novel mature size, so the size that we try to grow, and the colors will show which pre-adapted strategy they use. So the gray dots will be the cases where when we try to grow these sizes with small adapted strategy, as shown in here, whereas the blue one will be the cases where we try to grow these body sizes with large adapted strategies. So in the y-axis, I have this measure called fitness, And this is something we use to optimize in the genetic algorithm, but I'm not going to introduce the details of how this was calculated. But what I would like you to know is that this fitness was decreasing with body size, which is why I expected the results to look like this a little bit. So the small adaptive strategies to fail in large sizes and large adaptive strategies to fail in small sizes. So my first expectation was met. Small adaptive strategies did fail in large organisms. So if you look at here, for instance, after the null mature size of 100, most of them actually did not achieve any fitness. So it was 0. Whereas the other way around, like whether large adaptive strategies fail in small sizes, this expectation was not met. So large adaptive strategies could grow small bodies. As you can see, some of them actually fared similarly to the small adaptive strategy in small sizes. So there is an asymmetry between how robust these strategies are. So this implies that becoming smaller in size can be easier than becoming larger, because smaller organisms can use the strategies that are adapted to large size organisms, whereas vice versa is not true. And there's some evidence of this kind of things in the literature. For instance, this paper shows that maximum rate of evolution, when they look at the rate of evolution in the lineages going from small size to large size, so this increasing in body size took 10 times longer than the other way around, decreasing in body size in macroevolutionary time. So next question then was, so I've already shown that becoming larger might be tricky. So the question was then, can cancer risk constrain body size evolution? So this was my chapter number 3. Before I start with the details of the model and before introducing you to the results, I would like to talk about why we are interested in body size a little bit. So body size is one of the most important traits of the species that correlates with various aspects of life history. And it also might have advantages for within a species, like higher chances of survival, for instance, against predators. And it can also have a high, like larger individuals can have also higher fecundity and higher mating success. For instance, larger males having access to more females and things like that. So these advantages of large size is quite common in nature, and which is why there's also this trend that lineages tend to get larger in size over macroevolutionary time, which is called Cope's rule. Nevertheless, we are not really surrounded by very large organisms all around us. Like there are not like many species like elephants and whales around. And if you look at the mammal size distribution, you can see that. So here it shows that the body mass and the number of species with that body mass. And then you can see that smaller and mid-sized animals are way more common than very large ones. So there are various reasons why large animals might not be common, and these could relate to, for instance, higher extinction risk that is associated with large size, or resource limitations that limits lineage from getting larger in size. But as you can imagine, today I'm interested in talking about the role of cancer risk in all this. So we asked, can cancer risk constrain body size evolution? And used an analytical model and a simulation model in a complementary way to look at that. But today I'm going to only present you the results of the simulation model. So we took the endpoint of this chapter and we made use of these pre-adaptive strategies for a small size that were optimized in this chapter. So these were 4 different strategies. And we initiated 4 different populations with these strategies. And in these populations, now we assume there was sexual reproduction. So there were males and females. And we also looked at different scenarios. And you don't have to look at the scenarios right now. I'm going to introduce them one by one during the results. So the baseline result I'm going to show you now is when reproductive competition was settled by size. What I mean by that was probability of reproducing in both males and females depended on size, and large size was favored. And then I assumed there was no sex specificity in neither size nor growth strategy. So both sexes express the same size and same growth strategy. And growth strategy refers to this set of 7 traits that I talked about before. And the growth strategy was not evolvable. So once this population started with a set of traits and they did not change the traits as response to anything. So in the results, I'll show in the x-axis time and the y-axis the target mature size that is evolved. And you can see that with the size selection in both sexes, size increased in time. But this was not an open-ended increase, so this increase was limited. And more strikingly, strategies differed in their response to size selection. So for instance, if you look at strategies number 3 and 4 in here, the lineage that started with the strategy number 3 was much better at becoming larger in macroevolution time compared to the strategy that started, the lineage that started with strategy number 4. And this was actually in line with our results from the previous study as well, where we showed that the size threshold after which these strategies fail differ from each other. So even though these strategies can achieve the same fitness for the size that they are adapted to, they might differ in how permissive they are for size changes when we try to use them to grow a large body. So there might be some lineage-specific constraints to body size evolution. And this might potentially be one of the reasons why some lineages could become larger in size, whereas some others could not. And there's some evidence for this in elephants, for instance, where it has been shown that elephants had this pseudogene that became refunctionalized, and this refunctionalization coincides more or less with body size increases. So these researchers have this hypothesis that this might be permissive for them to increase their size. And this gene is related to DNA damage response, which is a part of this set of strategies that we look at as well. So I said so far we used a fixed strategy that did not evolve in time. But what if we let this set of strategies, with 7 traits that make part of the strategy, evolve with body size? So this is the next scenario we looked at. And co-adaptations in strategy allows a larger size increase, which is not surprising because it's something also I've talked about before, and we see in the nature that there is some in all these large size and long-lived organisms, there is some positive selection in cancer-related genes. And in here, we also see that co-adaptations of these strategies is essential for large size, evolution of larger sizes. So far, we assumed both sexes were selected for a large size. And now I want to look at this case when sexes might differ in their reproductive competition. And for this, I implemented another another way this reproductive competition can be happening. And I'm not going to go into details of how this budget was calculated, but what I would like you to know is that this budget selection favors small size. So if there is budget selection in both sexes, then the evolved body size is relatively small compared to the case where there's selection for size in both sexes. So with that, now I'll show you the results of when males were selected for size and females were selected for budget. And what we see is, well, first of all, body size increases. And it seems like size selection kind of overpowers to an extent the budget selection. But the increase in size is less than the case when both sexes were selected for size. So there is some different interest between the sexes, and this sexual conflict can constrain body size evolution. But so far, we assumed both sexes were expressing the same size, they were using the same alleles to determine their size. And then the next question is, what if size is sex-specific? And there are various examples of this in nature. There are a lot of species with sexual size dimorphism. So here I'm interested in modeling this. So again, there will be size selection in males, budget selection in females, but now I allow sexes to differ in their size. And as expected, we see the evolution of sexual size dimorphism. Males, shown in lighter colors in here, evolve to a larger size than females, shown in darker color in here, because males are selected for larger size, whereas females are selected for budget, which favors smaller sizes compared to the size selection. So what are the consequences of this sexual size dimorphism? So just to remind you, this is how the size trajectories look like. And now I will show you the sex differences in mortality causes. And in here, in the x-axis, we have time again, and the y-axis, there'll be % of different death causes. So these mortality causes are related to stem cell management and somatic maintenance, as I mentioned before, but also cancer and extrinsic mortality. And you don't have to look at all these mortality causes, but what I would like you to see is this cancer shown in red in here increases in time because males increase in size over time. And as males are larger than females, they have a higher cancer incidence compared to females. So larger sex has higher cancer incidence. And there is some evidence for this from the literature as well. Humans are a well-known example, for instance, when males, for most types of cancer, have a higher incidence. But there's also, for instance, some evidence from other organisms like this family of fishes where large size favors large size advantageous for reproductive competition, but also is associated with melanoma incidence. So then we wanted to look at what sex differences in size means in terms of life histories. And here I'll show adult population size, age at maturity, and lifespan. And in red will be the females, and in gray will be the males. And the x-axis shows different scenarios, but I will only focus on this scenario today, which is this body size evolution with sexual size dimorphism that I've shown before. So with this, we see that sex ratio is female biased in the adult population. So there are more females than males in adult population. And we see that age at maturity of males is higher than females, whereas average lifespan of females is higher than males. And if you actually look at the values of here, male average lifespan is actually shorter than age at maturity, which indicates that males had higher juvenile mortality, which helps explain why adult population size was female biased. So to sum up this bit, larger sex had smaller adult population size, higher juvenile mortality, and shorter lifespan. So I started this chapter asking about whether cancer can constrain body size evolution, but I haven't yet disentangled cancer from all these different death causes. So this is the purpose of this bit. So what we did in here was to remove cancer-causing mutations from the simulation. So we set the mutation rate, like oncogenic mutation rate, to 0 and wanted to see how body size evolved under this scenario. So in the x-axis here, we have the body size that had evolved under cancer risk, and the y-axis we have body size that evolved without cancer risk. And the diagonal, so above the diagonal are the cases where removing cancer risk led to larger body size, whereas below the diagonal is the cases where removing cancer risk led to a smaller body size. So you don't have to know what are the different scenarios in here, but I would like to show is we see the evolution of larger size when lineages were cancer-free. And this was especially the case in larger lineages, which was also in line with our analytical results that I haven't shown today. So there were a lot of results in this part. So just to sum up this chapter, lineages can differ in their response to size selection depending on their initial strategy. But co-adaptations in growth strategy can lead to larger increases in size. Next, I've shown that sexual conflict can constrain body size evolution. But if we allow sexes to express different sizes, then we see sex differences in mortality causes and life histories. And finally, I've shown that cancer risk can constrain body size evolution, which was particularly the case in larger lineages. So far, I talked about the difficulties of becoming larger in size, but there is also a possibility of lineages of getting smaller in size over macroevolutionary time. And this is what we're interested in the next chapter. So one famous lineage that got smaller in size over macroevolutionary time is the theropod dinosaur lineage that led to birds. So birds have evolved from relatively large dinosaurian ancestors over millions of years. And as I've shown in the introduction, dinosaurs also had cancer. So this implies that dinosaurs also needed to have they probably needed to have cancer suppression mechanisms as well. And since they are larger in size compared to their descendants that are birds, they might have had higher cancer suppression. And then the question is, how quickly these cancer defenses adapt during this miniaturization, the body size shrinkage process? Did the birds keep these defenses or did they lose them over time? We are also interested in birds because they live longer than similar sized mammals. So here in the x-axis, we have adult weight, and the y-axis is the maximum longevity, and in red is the bird and blue is the mammals. And you can see that birds live longer than mammals of similar size. And there are some interesting anecdotal examples of this as well. So for instance, this is this albatross called Wisdom, who hatched in 1951, who is probably older than most, if not all, the people in the audience today. And she actually still reproduces. So there are these examples of crazy long lifespans in birds. But coming back to the cancer aspect, birds also seem to have a relatively low cancer incidence compared to mammals and reptiles, which brought the question, could they have this high cancer defenses that have evolved in their dinosaurian ancestors because there was some lag and they didn't lose these defenses during their body size shrinkage? So I started this chapter by asking what happens when lineages get smaller in size, but more specifically, the question is, can evolutionary lags explain cancer robustness in birds? So I will introduce the model, and this model I will introduce a little bit more in detail, but I will try to walk you through it step by step. So first of all, I already mentioned that body size decreases in time. So this is what we assumed. And then there are two aspects related to mortality risks. One is that number of cells decreases. So I already said that number of cells has associates with cancer risk. So we assume that cancer risk decreases over microevolutionary time as dinosaurs get smaller in size to birds. Another thing that we assumed was extrinsic mortality risk increases. In the second chapter, I already talked about the fact that large-sized organisms might have some advantages in terms of survival probabilities for predators and things like that. So we assume that as organisms get smaller in size, they have an increased risk of dying from extrinsic causes. So in a nutshell, this probability of dying from these two different causes, the importance of these different risks kind of shifts while the birds get smaller in size. And with that, we calculate the probability of being alive and use it to get to lifespan. So this lifespan depends on cancer defenses in our simulations, shown in D in here. And what we do is we let mutations happen and changes cancer defenses and thereby change lifespan. And the question is, will these mutations fixate in the population? So will they lead to substitutions or they will not be selected? For this, we have to get the selection coefficient. So this is basically how better off in terms of fitness these individuals are using the new mutated defenses compared to their current defenses. So you don't have to know everything about this, how selection coefficient was calculated, but it depends on lifespan, cancer defenses, and costliness of cancer defenses. And what I mean by costliness of cancer defenses. So I already showed that this fish, for instance, had, if males had a large body size, they had higher reproductive success, but also higher cancer risk. So that's what we assume. So we assume the other way around, if defenses are costly, they might reduce reproductive success. So we implemented three different strengths for this trade-off. So if this trade-off is very strong, cancer defenses, this relationship between the decline in fitness by cancer defenses is quicker than if they have low cost. So this trade-off is weaker between cancer defenses and reproductive success. And with this, we use this to calculate rate of evolution, rate at which these kind of things will happen and substitute in the population, like these mutations will fixate in the population. And these depends things like effective population size and mutation rate per generation, which I'm not going to talk about today, but they also depend on different aspects of body size decrease. But what I want to talk about next is, so I already introduced the fact that extrinsic mortality rate might increase when the body size gets smaller. But one particular thing about birds is that they fly, and flight is usually associated with longer lifespan. So it might maybe, for instance, make it easier to escape from predators. And that's why we implemented another version of this model where we allowed birds to fly and this flight innovation to reduce extrinsic mortality risk at the point that it happens. And we selected these points basically sort of thinking about Archaeopteryx, which was one of the ancestors of the birds we see today. And I'll show the results for only one of these time points. So this will be a very big and complicated figure, but I'll walk you through it step by step. So in the x-axis, you have cost in here, this costiness, this trade-off that I mentioned before, and it goes from low to high. So when it's low, there's a weaker trade-off between cancer defenses and reproductive success. When it's high, there is a stronger trade-off. And the y-axis, there is the mutation rate. And then I will show 4 different scenarios in terms of body size change. So in the leftmost part, we have the lineage that stayed constantly large, so like dinosaur-sized, and did not fly. And in the rightmost part is something like mammals, a lineage that stayed constantly small and did not fly. And in the middle, we have 2 miniaturization cases, one with flight and one without flight. So first, I will show the number of substitutions that happened in these different cases. And this goes from gray, where there were no substitutions at all, and from yellow to blue and darker blue, it will indicate more substitutions. And each small pixel will correspond to a combination of mutation rate and cost. So to start with the baseline case, so these 2 cases start at their optimal defenses, but they still have some substitutions happening because of this mutation selection balance. So they basically fluctuate around the optimal defenses that they already have. But what I would like to show is that there is this boundary below which there were no substitutions. And this is what I will call mutational boundary. And this will be important for understanding coming results. So you can see that in birds, this mutational boundary was lower, especially when defenses were costly. So what does this mean? So there were more mutations happening at this area compared to these ones that didn't have any substitutions in that area, because the selection for losing costly defenses was strong. And you will see what I mean by this when I show you the defense plots, which are coming up next. So here I would like to first show you how the level of defenses change across costs values. So you can see that from left to right, the level of defenses decreased, because as defenses got more costly, the optimal level of defenses got lower. And in general, you can see like the dinosaur one, the dinosaur panel is more blue and darker blue compared to the mammal one. So the dinosaurs, because they were larger, they required more cancer defenses. And now we can look at the miniaturization cases. So here the mutational boundary comes into play. Here I already told you that there were no substitutions. So this parameter region first kept the dinosaurian cancer defenses. And above this mutational boundary, they evolved to the level of cancer defenses that were optimal for their body size and their flight situation. And so how does it connect to cancer probability? So this will show probability of death from cancer. So this color is in here. And yellow indicates lower probability of dying from cancer, whereas the darker blue indicates higher probability of dying from cancer. And I already mentioned that the level of cancer defenses was lower in this part of the parameter space because they were very costly. And accordingly, you can see that probability of dying from cancer was higher, and it was getting higher from low cost to high cost. And overall, dinosaurs had more probability of dying from cancer than small-sized mammals because they had basically more cells and more cancer risk. And now when we look at the case with the birds, above the mutational boundary, their probability of dying from cancer was kind of similar to the mammal-like lineage. Whereas below the mutational boundary, we see this case that the probability of dying from cancer was lower than both of the other constant size lineages. So you can see this as the predominance of yellow color in this part of the parameter space. So this is not to say that bird lineages were definitely below this mutational boundary. So it depends on what part of this parameter space the actual bird found themselves. But if they were below the mutational boundary, as I've shown in here, retaining dinosaurian cancer suppression might be the reason why they have lower cancer incidence. And what does this translate to in terms of lags? So by lags in here, I mean difference to the case where fitness is maximized. So this will be the lags in cancer defense. So how different the cancer defense that has evolved at the end of the simulations was from what would have been optimal for the body size and flightiness or flightlessness of the birds. And the same thing for lifespan. And for fitness, it will be how different the fitness was from the maximum fitness. So the blue colors will show higher than optimal, red colors will show lower than optimal. As I've shown before in the previous one, the birds have kept the dinosaurian cancer defenses below the mutational boundary, which meant that the defenses in this part of the parameter space was higher than optimal. And I've shown that they had lower probability of dying from cancer, which resulted in longer than optimal lifespan. So these all sound very good, like higher than optimal cancer defenses, not getting a lot of cancer, and having a longer lifespan. So what is the catch? And the thing is, when we look at the fitness that these lineages could achieve, below this mutational boundary, if they kept the dinosaurian cancer defenses, which might be costly, then they had lower reproductive success. So these long lifespan and higher cancer defenses came at the expense of reproductive success. Flight, to an extent, reduced these lags. So I already mentioned that flight might lead to a longer lifespan. So having longer lifespan meant that they could benefit from these defenses for a bit longer, so which meant that the lags in fitness was less. But nevertheless, it didn't completely compensate for having too high cancer defenses. So in some, birds might be too robust for their own good if they keep the dinosaurian cancer defenses. So why retain these costly cancer defenses? And one idea was that these defenses might have some pleiotropic effects. And what I mean by that is, I already told you that naked moras have this thing called early contact inhibition, and they use a special molecule for that, which the researchers think that might be co-opted from adaptations to underground living. So assume that for some reason naked moras don't need that much cancer suppression anymore, unless they find another innovation that allows them to continue underground living without losing this phenotype. It might mean that they might not lose this molecule. And if birds also, for instance, have this dinosaurian cancer defenses co-opted for another ecological function, as long as this ecological function is there, they might not lose these cancer defenses. So with that, I will start my discussion. And before that, I want to recap what I've shown through this presentation. First of all, we asked how are cancer defenses optimized across body sizes, and shown that there are multiple ways of achieving similar cancer defenses, and large adaptive strategies might be more robust than small adapted ones. Next, we asked, can cancer risk constrain body size evolution? And first showed that lineages might differ in their response to size selection depending on their pre-adapted strategy. But if we let the strategies evolve, we observe larger increases in size. So co-adaptations of cancer defenses and growth strategies might be essential for size evolution. And next, we've shown that sexual conflict can constrain body size evolution. But if we allow sexes to express different sizes, then we observe sexual size dimorphism. And this sexual size dimorphism might be associated with differences in mortality risks, as well as life histories. Finally, I've shown that cancer risk can constrain body size evolution, which is especially the case for larger lineages. Next, we asked, can evolutionary lags explain cancer robustness in birds? And then showed that if birds have retained dinosaurian cancer defenses, they might have low cancer risk and long lifespan, but this might come at the expense of reproductive success. So with this presentation, what I wanted to show you is that cancer is a thing that is a phenomenon that affects all diversity of life who are multicellular. So what I would like you to take home, like first point I would like you to take home from this is studying cancer across the tree of life can inform us about different cancer mechanisms long-lived and large-sized organisms might be using, as shown in the dinosaur example in this presentation, but as well as naked mole rats or various large-sized and long-lived organisms like whales, bats, and elephants. And the next thing I would like you to take home is studying diversity, having cancer in the back of your mind, might be also beneficial in understanding the patterns of diversity that we observe. And this is not to say that nothing in biology makes sense except in the light of cancer, but body size is one of the most important aspects of species life history, and it associates with cancer, so it might be useful to keep cancer in mind when studying body size evolution and related traits. So with that, I'm at the end of my talk, but I would like to thank a few people. First and foremost, to my supervisor, Hannah, for many years of support, both scientifically and also for her understanding and support during personal difficulties. To my PhD committee members, Natasha, Wolf, and Kathleen, as well as my external reviewer, Jean-Michel. I would like to thank URPP Evolution in Action for funding and University of Zurich. I also collaborated a lot and talked a lot with people from Arizona Cancer Evolution Center, who will also be funding my postdoc in here until the end of next year, like this year. And especially to Mark, who was one of the people who was co-author in one of my chapters, as well as various people who I could not fit in this slide. And I would like to thank all the coconuts, past and present. There's no way I could capture all the people in one picture, and I don't think I even have everyone in here, but I cannot imagine a better group to work with. It was very enjoyable, both like fun times, but also scientific discussions. I would also like to thank all the URPP and Department of Evolutionary Biology in UZH, all the people in there. I would like to thank Turkish Society of Ecology and Evolution for their invaluable work promoting evolution in Turkey, despite all the obstacles, but also I found this PhD position through their mailing list, actually, with this cascade of random events, which was also very nice. And I would like to thank all the other collaborators I had during the PhD, Thomas, Matthias, Paco, and Josh, and my past supervisor, as well as Mim, for kicking off my career path as an evolutionary biologist, and these supervisors for providing me support during my master's and also later in life. And of course, I would like to thank many, many, many friends for all the enjoyable times and fun times we had, and as well as your support during bad times as well. And I would like to thank to my family, and I was very privileged and lucky to have very supportive parents growing up. Unfortunately, my father couldn't be here because he was diagnosed with cancer at the beginning of my PhD. So I have a personal relationship with cancer as well. So he's not here today, but I would still like to thank him for everything. And I would like to thank to my mom and my sisters for their support and love during difficult times, and yeah, making me feel like I could do it despite everything. So I haven't talked much about the details of all these chapters, and all of them are online actually. So you can, if you're technologically minded, scan this QR code and go to my Google Scholar and read the details. And there will be some sort of virtual get-together afterwards at around 6 to see what happened at the end of all this. And you might already have the link, so you don't have to worry about the meeting ID if you have the link. But if you don't, you can use this meeting ID to access that. And yeah, with that, I'd like to thank everyone for listening and joining today.